tell you some story about the application of mass spectrometry after you understand its history and how it works. The flourishing and developing tree of mass spectrometry still sits out to early fields and make fruitful gains. There are many researches related to biology field using mass spectrometry. Let's go back to the 20th century. We begin to use mass spectrometry to characterize molecules, say identifying organic compounds in 1956 and start peptide sequencing by it in 1966. In 1989, ESI on biomolecules began to germinate as one of the most important discoveries in mass spectrometry that time, and monitoring enzyme reaction with ESI-MS occurs at the same year. In 1992, Donhart set the standard with low-level peptide analysis. In 1993, protein mass mapping, and which also called fingerprinting, turns into one of the most important applications of mass spectrometry. MS of a virus then demonstrates the viability of a virus after passing through a mass spectrometer in the amazing 1996. In 1999, quantitative proteomics and metabolomics with isotope labels becomes popular. Now back to the 21st century. There are shotgun lipidomics focused on uses analytical chemistry into lipids in 2003, and fancy surgical intelligent knife detecting cancer cell in 2013. What's more, drafts of the human protein utilizing of high resolution and high accuracy Fourier transform mass spectrometry in 2014. Today, we are focused topics about mass mapping and surgical intelligent knife and shotgun lipidomics. Let's talk about protein mass mapping, which is also called fingerprinting first. It is an analytical technique for protein and peptide identification developed in 1993 by several groups who briefly introduced its procedure, which is called PMF workflow. Unknown protein is first clipped into smaller peptides. We measure absolute masses with mass spectrometer as the next step. Masses are then compiled to database to find the best theoretical match, and we can get our unknown protein clear intensification. Cannot follow it? Let me bring more details in it. It has three general steps: sample preparation, mass spectrometric analysis, and computational analysis. Sample preparation aims to get peptide segments from sample proteins. We need to isolate proteins first through SDS page. Load sample to stacking gel firstly and connect it to the electrode. Run, then protein separate according to its mass. By cutting out gel spots, we can get the protein we want. Add protease like trypsin, chymotrypsin, or V8 protease into the sample to clip it into a peptide. The protein then magically turns into small pieces. What is the mixture of peptide looks like? The blue strings on the right-hand side shows the abstraction of triadic peptide mixture. Every peptide has a basic C-terminus. Of course, it is more messed up than it seems to be. Peptides mixture then get extracted with acetonitrile, dried under vacuum, and dissolved in a small amount of distilled water. After extraction, we continue on mass spectrometric analysis. We can get higher sample throughput by MALDI-12 or ESI-12 and measure small fraction of peptide by MALDI target. It is an easy step to add matrix, a chemical that absorbs light. Compounds get wrapped within the sample plate. Matrix and peptide molecules co-crystallize on the MALDI targets. This machine is a MALDI-12, which I found on the internet, by the way. Then we go to the computational analysis part. The peptide masses are compiled to either a database containing no protein sequences or even the genome. How to compare it to genome? This is achieved by using computer programs that translate the known genome of the organism into proteins. There follows comparison. How are they get compiled? Thanks to technology. Software programs like MSFit, Mascot, Peptident, and Profound cut all these proteins into peptides with the same enzyme used in the chemical cleavage. We can then compare between the peak list of major peptides and all theoretically calculated peptides. Possible matches will be returned in a result table. Then we found our most ideal match and identified the unknown proteins. Overlook of the whole workflow. Sample cut from jaw is digested by protease. Extraction combined with matches and measured by mass spectrometry. The results are then compiled to database. Sample get identified if there exists perfect match in database. Here is an example for the protein mass fingerprint of ADH peptide digest and creatine phosphokinase peptide digest. We could get the information about the intensity and sequence from the pick list figure. Possible matches show with score and rank, and return in a result table with some parameters like number of peptides matched, etc. Fingerprinting, of course, is useful but not unlimited. It has the advantage that only the masses of the peptides have to be known and peptide sequencing is then unnecessary. It also has limits, such as the protein sequence has to be present in the database of interest. Most PMF algorithms assume that the peptide comes from a single protein. The presence of a mixture can significantly complicate the analysis and potentially compromise the results. Now let's talk about surgical intelligent knife. It's a device that connected to an electrosurgical knife attached to a cancerous tissue invented recently in 2013. It can tell surgeons immediately whether the tissue they are cutting is cancerous or not with high accuracy that normally takes up to half an hour to review using laboratory tests, and it is invented by Dr. Zoltan Takas from Imperial College London. As Fomo said, is based on electrosurgical knives, which applies an electrical current to rapidly hit tissue, cutting through it while minimizing blood loss. We can see the electrode connected to electrosurgical unit and atmospheric interface and is doing something by contacting the patient. Now let's talk about shotgun lipidomics, which uses analytical chemistry to investigate the features and significance of alternation in lipids and protein constituents, mediating biological function in cells. It is fast, high sensitive, and can identify hundreds of lipids missed by other methods. And we have developed many methods combined with MS to investigate different kinds of lipids. We take steps to extract lipids, infuse sample, ion scanning, and then do lipid identification and quantification. Traditionally, mass spectrometry, as an essential part of the analytical instrumentation market, has been used for routine tests in biotechnology, pharmaceuticals, food safety, petrochemicals, and environmental analysis. It provides highly specific molecule information about excellent sensitivity from determination of atomic mass and isotopic abundance to identification for organic compounds, from study of electron collision to satisfy demands of petrochemical industry. It is such a good technology but is still facing challenge in size, functionality, and budgets. It usually consists of a large box with a footprint that can easily take up to 6 feet bench top and typically performs one task slowly with a retention time ranging from 5 minutes to 10 hours. The cost of the machine is between $50,000 to $120. According to its limits, We could improve this technology in the future by shortcut of task time, improve faster throughput, smaller footprint with more precision, integrated online automation using cloud computing with core laboratories to magnificent and improve mass spectrometry software to make analysis of data easier. And there you have it, a basic overview of MS applications below. And stay tuned for our future videos. Thank you.